Hello again. Now we're on part B of chapter 3, and this is going to be looking more closely at ways that substances are transported across the cell membrane or plasma membrane. First of all, there are active processes. There's two major types of active processes. There's active transport, and then there is a design called vesicular transport that involves vesicles. Both of these will require ATP because the solute is either too large for the channels, or it's not lipid soluble, or it needs to move up its concentration gradient. Active transport involves carrier proteins. These are typically called solute pumps. These will move the solute against or up the concentration gradient, therefore it requires ATP, energy. Primary active transport. A great example of this is the sodium-potassium pump. It's the most familiar, and we will be talking about this when we get to muscle contraction and the conduction of nerve impulses in the nervous system. The carrier, or the pump, is actually called the sodium-potassium pump, and the enzyme that it uses is the sodium-potassium ATPase that hydrolyzes ATP to provide the energy for this pump to function. This pump is located in all plasma membranes. The sodium-potassium pump, first of all, understand that there are actually some sodium and potassium leakage channels that are always going to allow slow leakage down each of these ions' concentration gradients. So when sodium is highly concentrated on one side of the membrane, it can leak through these channels down its concentration gradient. And potassium also has a leakage channel, and so it can diffuse down its concentration gradient through the potassium channel. So the sodium-potassium pump is an antiporter. That means it pumps against the sodium and potassium gradients in order to maintain a high intracellular potassium concentration that's inside the cells, and also a high extracellular sodium concentration right outside the cells. This figure is actually, it has a step art, so I'm going to go through this. It starts out when 3 cytoplasmic sodium ions, and this should be a plus right here. It's too tiny to change it. 3 sodium ions, intracellular sodium ions that is, are going to bind to this pump. Those are sodium ions inside the cell. The sodium binding actually promotes the hydrolysis of ATP, and the energy that's released during this reaction will phosphorylate the pump, basically activates the pump. So this phosphorylation causes the pump to change shape, and it will expel those 3 sodium ions to outside the cell. Now 2 extracellular sodium, and this should be a plus right here, potassium is also, potassium and sodium are both positively charged ions. So there's 2 extracellular potassium ions are going to bind to the pump, and then the potassium binding will trigger the release of the phosphate, and now this dephosphorylated pump will resume its original conformation. So in other words, the pump is going to take 3 sodium ions from inside the cell and pumps them out, and then it will pick up 2 potassium ions from outside the cell and release them to the inside of the cell. These were also a little out of sync, but anyway, you could get the idea here. Keep in mind, sodium and potassium are cations. It should have a little plus on them. So secondary active transport. There's different types of secondary active transport, and it gets actually much more complex than this. Co-transport, though, would transport more than one substance at a time. A symport system transports the substances in the same direction. An antiport system will transport those substances in opposite directions. This is showing a primary active transport right here with that sodium-potassium pump. Now they have the little pluses there, which makes me happy. Okay, so the ATP-driven sodium-potassium pump stores the energy by creating a steep concentration gradient for sodium entry into the cell. So in other words, when you are using these pumps, it ends up with having a high concentration of sodium right outside the cell, and there will be a high concentration of potassium inside the cell. This is showing you an example of secondary active transport, and this is, it's actually when sodium, this is a symport transporter, when sodium is transported across the membrane, it also is going to drive the transport of glucose. Vesicular transport. This is a different type of transport, and what this does is it can transport large particles and fluids across the plasma membrane in vesicles. Remember, vesicles are little membrane-bound sacs. This does require energy. It requires ATP. And the functions for vesicular transport, and you may have heard of these before: exocytosis, endocytosis. Remember, exocytosis is the cell spitting things out, removing things from the cell. Endocytosis is when the cell takes substances in, and this includes phagocytosis, pinocytosis, and a very specific type called receptor-mediated endocytosis. Also, there's something called transcytosis, where a substance can be moved in a little, it'll be packaged in a little vesicle and move across from one side of the cell to the other. Endocytosis and transcytosis. These actually use protein-coated vesicles. Often the receptor is mediated and very selective. And again, transcytosis is when a substance is moved across but within the cell. Endocytosis includes phagocytosis, which technically means eating by the cell. Okay, the cell is eating. This is used by macrophages and some other white blood cells. So you may remember the macrophage is a white blood cell that will actually engulf foreign subs like a bacterium or some kind of debris in the body, and it will just then fuse with a lysosome, which will break apart that invader. Phagocytosis is a way to take substances into the cell. And this is showing you phagocytosis, and the cell is actually engulfed the large particle by forming pseudopods, which are called false feet, around it, and encloses it in a membrane called a phagosome. The phagosome actually combines with a little vesicle called a lysosome, which contains digestive enzymes. And so this becomes a phagolysosome, actually, and the undigested contents are going to remain in the vesicle, and they can break. Actually, the lysosome will break these apart, and then they'll just get rid of the debris by exocytosis. Endocytosis. Another form of endocytosis is pinocytosis. This is when the plasma membrane folds in, and it can bring extracellular fluid with any of the dissolved solutes that happen to be there inside the cell. This is used during nutrient absorption from the small intestine by the cells lining the small intestine. Pinocytosis, this is the cell actually gulps a drop of extracellular fluid containing solutes and forms a little vesicle. No receptors are needed, so this is pretty nonspecific. although most vesicles are protein-coated. They are learning more and more about these processes every day that we speak. Receptor-mediated endocytosis allows a specific endocytosis, so it's going to be bringing something into the cell that is very specific, something that the cell needs, and also transcytosis. So it will take a very specific material that will initiate this, the receptor. So it will activate this receptor, and when this vesicle is formed, it can move the material that are inside of that vesicle across the cell. So both specific endocytosis and specific transcytosis. Exocytosis. This is usually activated by a cell signal or a change in membrane voltage. The substance will be enclosed in a secretory vesicle. Remember, this is not talking about it being a secret, but that it's secreting something. Usually when we use that word secretion, it's talking about something is going to be sent out by a cell or a group of cells secreting something. So the secretory vesicle will actually contain something that it will then release to the outside of the cell. So where do we find this specifically occurs? This would be with hormone secretion. So endocrine cells will produce and then secrete the hormone through using the secretory vesicle. Neurotransmitter release as well. So the neurotransmitter chemical will be stored at the axon ending of a neuron, and then it will be, when it receives the signal, it will actually release it into the synaptic cleft. We'll get to that in more detail with the nervous system. Also, mucus secretion and ejection of wastes by the cell are undertaken through this type of exocytosis. And this is showing you the step art, and this is quite a bit of detail showing you the process of exocytosis. Go back to that. This is showing you that there's here's the plasma membrane, and they've actually discovered these little complex of proteins. This is called a snare, and it actually will signal the secretory vesicle here that responds to that, and it will actually be carrying this molecule that's supposed to be secreted, so it will be guided by this snare right here. The membrane-bound vesicle migrates to the plasma membrane, and this fusion pore will be formed. The vesicle and the plasma membrane fuse, and this opens up a pore. The vesicle contents are then released to the cell exterior, and and that's the end. Okay, so here, actually, let's go this way. Beep! Figure 3.14 is exocytosis step by step. The membrane-bound vesicle will migrate to the plasma membrane, where it will find one of these plasma membrane snares or T snares. Then the vesicle and the plasma the proteins here first will bind to the T snares, and then the vesicle and the plasma membrane fuse, and a pore opens up, and then the vesicle contents are released to the cell exterior. Here's a photomicrograph of a secretory vesicle releasing its contents via exocytosis. This is an electron micrograph using an electron microscope, so it shows you that they're not just making this up. They actually can see this process using an electron microscope image. Table 3.2 gives you a summary of active membrane transport processes. Primary active transport, the energy source is going to be ATP, and so this is the transport of substances against their concentration or electrochemical gradient. It's performed across the plasma membrane by a solute pump directly using the energy of ATP hydrolysis. The examples would be moving the ions sodium, potassium, hydrogen, calcium, and others. Secondary active transport uses the energy of the ion concentration gradient that was maintained, that is going to be maintained with ATP. Co-transport, coupled transport of two solutes across the membrane. The examples would be the movement of polar or charged solutes such as amino acids, calcium, and hydrogen ion. This is showing you endocytosis. Another way of, these are vesicular transport processes. There are quite a few types right here that are summarized for you, and you can look at this in your book or on your PowerPoint in more detail. And then this would be endocytosis, taking materials into the cell, and then this is using a vesicular transport for exocytosis, taking substances from inside the cell to eject them to the outside. Now, this is a discussion of the generation of the resting membrane potential. We are going to come to this in detail when we talk about muscles. And then near the end of the semester, we talk about the nervous system. Both muscle cells and neurons in the nervous system are considered excitable tissues. They will respond to a signal that's going to cause something to happen. With neurons, it would be the release of neurotransmitter that will send a message either through a sensory pathway or a motor pathway. With muscles, it would be when that muscle cell receives the signal, it will initiate a contraction, a muscle contraction. So, the resting membrane potential, RMP, is produced by the separation of oppositely charged particles, or voltage, across the membrane. Voltage just sounds like electricity. Well, it is. Ions it's an electrical phenomenon because, remember, positively charged ions or positively charged particles are going to be attracted to the negatively charged particles and vice versa. When you separate these opposite charges across the cell membrane, now we can say that the cells are polarized. Voltage, and this is actually electrical potential energy caused by the separation of opposite charges. It's only going to occur at the membrane surfaces. The resting membrane potential of cells will range from -50 to -100 millivolts in different cells. The membrane will be negative on the inner surface relative to the outer surface. So what maintains this? The active transport of the sodium and potassium ions right here is going to maintain excuse me, electrochemical gradients. The sodium-potassium pump is going to continuously eject 3 sodium from inside the cell to the outside, and then it will bring in 2 potassium ions. Neuron and muscle cells actually will upset the resting membrane potential. And this is going to, now we call this creating an action potential by the opening of gated sodium and potassium channels in response to some stimulus. Sodium leakage channels are closed and some potassium leakage channels are open. To establish the resting membrane potential, remember, there is some diffusion of potassium from inside to outside across the cell membrane, and that's actually what's going to establish this resting membrane potential that's negative along the cell membrane's inner surface. So I believe there's a step art. Oops, it's not. Okay, so the key role of potassium in generating the resting membrane potential. 1. Potassium will diffuse down their steep concentration gradient. Remember, the potassium has been basically dammed up in here, but it's pumped up into the cell through the sodium-potassium pump. Sodium is going to be pumped out. So you have a lot of potassium here, and it's actually always going to be kind of leaking out of the cell via leakage channels. The loss of potassium from inside the cell is going to end up It actually kind of draws these negatively charged proteins to the surface or the inner surface of the cell. So this is going to be really negative along the inner surface. The potassium also move into the cell because they're attracted to the negative charge established on the inner face of the plasma membrane. A negative membrane potential of minus 90 millivolts is established when the movement of potassium goes out of the cell, and this is equaling the potassium movement into the cell. At this point, the concentration gradient promoting potassium exit exactly opposes the electrical gradient for potassium entry. Just keep in mind here that the movement of the potassium going out is going to allow this surface to be very negative because it's leaving, and all these sodium ions out here, plus those potassium that have leaked out, it's going to establish on the outer surface of the membrane to be very positive. We are going to come back to this later in more detail when we get to muscles. But selective diffusion will actually establish the resting membrane potential and also that sodium-potassium pump. Keep that in mind, and that's what we will be discussing later with the muscular and nervous systems. Okay, there's a few other kinds of cell proteins, cell adhesion molecules. There's thousands of cell adhesion molecules on every cell in the body. What do these do? Cell adhesion molecules will anchor that cell to extracellular matrix and to each other. It's really important that our cells stick together and tissues and organs stay in the right place. And that is partially due to these cell adhesion molecules. Plasma membrane receptors. These are receptors, and this is called contact signaling. It requires cells to touch each other to allow recognition of other cells. Scientists have realized that cells can actually touch other cells. There's little receptors that allow them to know, oh, I'm right here by other cells of my type. There's also chemical signaling. This would be the interaction between receptors on the cell membrane, receptor proteins, and what we call ligands. It's a fancy name. It's just referring to some kind of a chemical, such as a neurotransmitter, a hormone, or some type of a paracrine, which is similar to a hormone, but the paracrines do not travel throughout the bloodstream. Hormones do. So chemical signaling would involve the receptor on a cell that's activated by binding of some kind of a ligand to alter activities of that cell. And we will get to that particularly during the endocrine system. This is illustrating how a G protein acts. I'm just going to skip through this right now because I find it more complex than it actually is. And we'll be talking about it when we get to endocrine. And that is the end. Have a wonderful day, and I will see you with Chapter 3, Part C.